Thanks very much for that talk. That was great. Is there any studies on the incidence of mammary cancer in depending on the level of carbohydrates fed? Was there not a significant bit of research there recently? I think it was in dogs on tumor growth, where they dropped the cancer, or they dropped the amount of carbohydrates fed, and the tumor growth decreased. Or am I confusing that with something?
Sorry, Anna, repeat the question, please.
I can repeat. I can repeat the question. Connor is asking whether you know of any studies that know about the amount of carbohydrate in the food and the prevalence of cancer.
Yes, the cancer is not indicated the diet about the carbohydrate, but in nowadays the more important the kind of the diet, it's important the balance, because more important the modulated a diet and understand the form the diet the food interfere in different pathways in carcinogenesis.
Okay.
Okay.
Thank you. Thank you.
Okay. Is there any possibility that you can stay with us when we have this panel discussion a bit later today?
Okay. Yes.
So if you're going to, then we will have more time for questions. Can you see that you're sharing? Yes, you are. So the ones that are on Zoom, can you see that there is a dog with pink glasses on at the moment? Any yes or no?
Wait 1 second. I'll check the chat.
Yes. Okay, perfect.
Great.
Thank you very much. And so I will actually then present our next speaker, which I presented to you like 20 times already today, because he's my half of my brain. At least everything that has to do with IT, which is actually totally non-existent in my brain. So Robin has been with us now for several years, because he was also doing his master thesis with us on metabolomics after a diet intervention study that was done on dogs that were either healthy or atopic. And now he is working on a data set that comes from dogs that are either healthy or have some type of tumors, both cancers and non-malignant tumors. And also, as I said before, he is in the very good care of both me and Alessandra. Yes. So we will see to it that he gets his PhD done before the next century. Super. Okay. Thank you very much, Robin. It's yours.
Thank you. All right. I'm just checking that everybody can hear me. Yes. Good. Nice. Okay. Hi, everybody. And I'm going to be picking up where Alessandra left off. She had a bunch of background literature information, and I'm not going to really be talking much about background information, because when it comes to mammary cancer in dogs and diet, anyone want to venture how many studies have looked at what happens in the metabolism? Yep, that's correct. So this is definitely looking, you know, I have a picture here of a dog, and if you can see, there's something very strange with it. It's actually not a dog. It's artificial intelligence conception of a dog. It's not. It's this is done with ChatGPT. In any case, the point is just that we were just talking about how we can, you know, understand humans through studying dogs, and that's because dogs live short lives compared to us humans, and as a result we can track the progression of how a cancer develops and what it means, what the implications are, quite well, because, you know, we're alive to see the progression happen. But on the other hand, what we have interest in traditionally is human breast cancer. So we have a lot of information about human breast cancer. So it's actually like this interesting reciprocal situation we have, where our knowledge about human breast cancer so far is fueling our research on canine breast cancer, which in turn will help us understand human breast cancer. So there's very much this like One Health type approach to the problem. What I'm going to be talking about today is basically what I've been doing, which is try to understand how cancers in mammary tissue express themselves differently. And what I'm looking at is a non-invasive way of seeing that. So I'm studying urine samples. And I presented some preliminary data already a year ago, and I now have finally gotten a lot more information about both the metabolites that I'm studying as well as the dogs as I'm studying, because as you may recall from last year, a big problem was that yeah, I had all this raw data, but I didn't actually have the background information about those dogs that was kind of important. So in any case, I kind of, yeah, have established that dogs, like humans, have mammary cancers, and that, well, you know, thanks to Alessandro's work with me in the last year, we were able to go through the actual pathological reports from when the tumor was removed, and we were able to establish what kind of cancer we're talking about. And in many cases, we were able to differentiate with cases between dogs that had, say, a malignant tumor, dogs that had a benign tumor, and then dogs that had both. They were previously classified as just having a malignant tumor, but there's a big difference between having a malignant tumor and 3 different kinds of tumors. And well, if you're trying to establish a clear signal between one type of tumor and what you're going to expect to see in the urine if you're studying it. So a lot happened last year, and thanks to Ale, we sat down, especially last spring, and like worked every day on making sure that we were able to actually properly diagnose this, because, you know, shit in, shit out. If you don't have the proper like labels on the data that you're studying, you're not going to actually learn anything. But, you know, some things are already known about mammary cancer in dogs, and that is that, well, there are some risk factors. There are basically, you know, purer breeds are said to have a slight larger risk of developing it. And then also, you know, smaller dogs, I think that this also has to do with age, because it's that age onset that is very important. Modifiable factors have been studied, including in dogs, but diet hasn't yet. It's just the way things are. So let's see. Yeah, well, yeah, so what we can turn to is what we know about humans. And what we know about humans is that there are actually metabolic signatures of mammary cancer, and they include amino acids. And we actually named some amino acids here, like tryptophan and tyrosine. And they're also diet related, as you may recall from previous talks. And so that I really like makes us think this is actually worth studying. But the 2 overarching questions are that, can different types of canine mammary tumors cause different signatures that are detectable in urine? And could dietary influences on the metabolic health of infants, could dietary influences on metabolic health also influence the mammary cancer outcome or progression or malignancy? Basically, well, not only is it difficult for us to diagnose a type of mammary cancer if we don't know what the dog has eaten. If we know what the dog has eaten, does it make that picture easier? And then also, is there a trend? Like, we noticed that, okay, yeah, there's like a trend of, okay, we're starting to learn a signature of malignancy, say, in metabolites. And then we see that, oh, that trend has something to do with diet. It's very strongly correlated with diet. Well, that's interesting information, right? Yes. So in any case, what we're trying to do here is analyze urine samples. And what we're doing is basically just throwing them in a mass spectrometer and see what comes out. That's a type of study called untargeted analysis. We're just putting everything in there. We're just putting the sample in there, and we're going to see what this device can detect. And so we're using a mass spectrometer. We're using liquid chromatography to separate the samples, and I don't really need to go into that now. But in any case, that's then produces a lot of raw data. Somebody has to go through all that raw data, and then sooner or later you get some results, and then you can discuss them. So what we had was dogs. We had about 134 dogs when we started out, and depending on the research question that we're asking, and the models that I'm going to be showing you, I'm going to be showing somewhere between 78 and 117 dogs. So that has to do with the background information that I mentioned. So I can, as you can see, there's the first questionnaire, which was sent out when those samples were made. Sorry, when the samples were made and collected. And then, 82 respondents of the 134 were able to give an answer, but then it took some time to get all of the people who had participated in the original study to answer. It took about 4 or 5 phone calls in some cases. But in any case, we got a pretty nice group, and now we have this nice background information and getting a good idea of what their lifetime diet has been. And I will go into that in just a bit. But yeah, their diet basically is, a spectrum between kibble and raw, and then we also include home cooked and processed foods. And so those were then thrown into the mass spectrometer as I diagnosed. And now we can start looking at results. Now, this was the original data that we had. And I hope that you guys don't see some big trend here, because the point is the exact opposite. When we were using the labels malignant or benign, or now malignant and benign, because some of those malignant dogs actually also had benign tumors, we can see that with principal component analysis that we just like throw all of the data into our model, they're quite overlapping. This is very, very difficult to differentiate. But what if we differentiate them with the new groups? And I am going to actually move this thing. I wish I could make it disappear. Hide panel, hide. Floating meeting controls. Yeah, there we go. Nice. Okay. Yeah, I don't know if you can see up here. It's a little bit small. Apologies for that. But here we now have a list of 8 different types of tumors, or well, 7, and then you have the control group. The control group is pretty large. It's about half of all of the dogs, but that's fine. We're basically trying to establish what signals are different between the tumors, or dogs with tumors, and then healthy control dogs. And what we can see here is that if we differentiate the actual types of tumors, we actually get meaningful data. And there's actually quite a lot that's going on that's different. And so we can see actually that, yeah, there's about 500 of these like features that are different. And as a result, they're not as overlapping anymore. And hopefully you can see in this principal component analysis that not only is it explaining a lot more of the variance, there was only a few % in the previous one, but they're actually kind of separatable. Well, what if somebody would actually go through all of those features and then confirm that what we're looking at, a metabolite, is actually the metabolite that we're interested in? Well, the only way to do that currently is by hand. And you can semi-automate it with artificial intelligence, which I did, but that still left me with a list of something like 5,000 to 7,000 features, which I had to just by hand look at each spectrum, and it took a while. But it allowed me to create a list of things that, so what that meant is that I was actually able to establish a list of about 230 metabolites that not only we know about, but we were able to detect and confirm in these urine samples. And here we can see that, well, this is the diet portion of the talk. You can see that we have classes, and these classes are what I ended up going with, because this is where I was starting to see data. And I'm not going to go into the other ways I sliced and diced the data, but I noticed that there is some interesting stuff happening right around this 50 to 100% raw point. So basically, we had a lot of dogs, and we were asking them what they had eaten in their adult life. And we excluded dogs that switched around their diets a lot. So we were basically just really interested in dogs that were eating more or less the same thing, at least 6 months prior to sampling, but preferably their entire life. And we asked about that. In any case, the point just being that there were some dogs that ate 80 to 100% kibble. And then we had some dogs that ate 10 to 50% raw, and that could include kibble. But then the 50 to 100% raw is a predominantly raw diet. And the mixed, not raw, is just a varied diet that includes other types of processed foods as well as home-cooked foods. Yeah, and as you can see here, we have some interesting stuff where, for example, like vitamin B6, yeah, a heat map, red up, blue down. So we can see, for example, that 4-paradoxic acid, which is the downstream product of vitamin B6, for example, is significantly higher in the raw food group. And very interestingly, it's lowest in the tryptophan, which I mentioned earlier, is by far lowest in the dry food group. And then it was sort of like a little bit higher in the raw food group, but then highest in the mixed and the 10 to 50% raw group. Yeah, and then also you can see here that if we're able to quantify our results, we can also see some cool stuff like, for example, deoxycarnitine, which is one of the carnitines, we were talking about them earlier, are able to be measured, and you can actually establish that, okay, yeah, in the kibble group, they're significantly lower than in the 10 to 50% raw group. And then in the 50 to 100% raw group, you can see even higher amounts than in the 10 to 50% raw group. And this is exactly what you want to see. Yeah, and then you can see here in tryptophan, for example, I mentioned earlier, then you have the lowest amounts, more or less, in the, well, statistically speaking, the lowest amounts are in the dry group, though there is 1 or 2 in the mixed group that are also quite low. Yeah, well, the diet hence can be separated quite well in this study. And yeah, from another heat map, we can see that there is actually a lot going on due to diet, more so than what was able to be observed when looking at the cancer types. We can see that there's this cluster here. You can see here, the blue is my 50 to 100% raw feeders group, or raw eaters. And there we have some that are up and others that are down. And this is interesting. These are unannotated, unidentified features, but this means that there's actually some really interesting stuff worth exploring. And yeah, and we look at the diet again, yeah, we can see that there's a lot of overlap in the diet groups, except for in the 50 to 100% raw. And now this looks different because this is not the annotated small list of features, but this is just all of the features that we're looking at, which was about 60-something thousand. Okay. Yeah, and okay, well, yeah, there we were able to see, like, this is just another picture of the heat map, that some significant differences were seen due to diet. And then, yes, then we have the heat map for the different types of cancers when looking at all of the data. And we were saying, okay, yeah, there's actually quite a nice trend when you're looking at the unidentified or unknown compounds. And then, well, what if you combine it? Then you can see that, well, actually, out of those, there are some that overlap. There's an interaction between something like 24 of them. Now, that's really interesting because that's something that is significant due to both diet and cancer diagnosis and tumors. So if we took a look at one of them, for example, here I'm showing the the actual, or excuse me, the tumor diagnosis, we can see that arginine, which has been implicated in breast cancer in humans as a as an interesting therapeutic target, actually can be detected. And there's some kind of a trend here as well that we can see that we were talking about ductal, intraductal carcinoma as being like the most lethal. And there you can see that this is on a log scale. So we can see that arginine concentrations are somewhere in the vicinity of 50 to 100 times less. And that's interesting. That's meaningful. So what are the conclusions and take-home messages? Well, clear differences have previously been seen due to diet, and I presented that briefly last year. But what we were also able to see was that, unfortunately, the biological metabolites are the ones that we know about. The weird ones, like these, how would I explain weird? Exogenous metabolites, basically like these weird toxins, mycotoxins were brought up, pesticides, microplastics, etc. Those are all unidentified so far. So they could actually be explaining why we see such big differences in the unidentified groups versus the identified ones. The identified ones are basically well, metabolites. And they're known. They have some function in the body. But yeah, in any case, I'm running out of time already, so please ask some questions. But without further ado, I'd like to just thank Alessandra a lot, as well as my other supervisor at the University of Eastern Finland. I also want to thank the people who are supporting the R programs that I'm using, as well as the DogRisk group and supporters and funders. Thank you very much for your attention.